In this third and final video, we're going to complete our discussion of the alimentary canal organs, now beginning with the duodenum and running to the rectum. In the first part here, we need to look at the corresponding structures associated with the small intestine, the liver, the gallbladder, and the pancreas. In this image here, you can see first off the stomach, and from that, you can see how the alimentary canal continues as the small intestine. There's three regions here: the duodenum, the jejunum, and the ileum. The muscularis of all of these regions is bilayered, going back to the longitudinal and the circular components that we've seen in the esophagus and in the pharynx. It utilizes peristalsis for propulsion, segmentation for mechanical digestion, and then as it ends, it forms the ileocecal valve, also sometimes called the ileocecal sphincter, right here with the cecum of the large intestine. The majority of it is covered by visceral peritoneum, but the duodenum is going to be retroperitoneal, and that's where you'll see adventitia covering parts of the small intestine. And mucosa is quite developed, much like what we saw with the stomach. You're going to see many different cell types. You're going to see big crypts or pits, essentially. But even more than that, you're going to see how there's a large increase in surface area here of the small intestine. Now, this is important because one of the primary roles of the small intestine is absorption of nutrients. So in looking at this image here, you can see this zoomed out version of the small intestine. The first thing you see is that there are circular folds right here. So these are very large protruding regions here of the small intestine. So again, circular folds. And if you get a little bit closer to each one of them, what you'll find is that there's finger-like projections within these circular folds known as villi. Now, if you zoom into each villus, this is what you see. So here's those individual finger-like projections. In between them, you're going to see these intestinal crypts, much like this one right here. So this resembles your gastric pits from the stomach. But then in addition to that, all the villi themselves are made up of tiny little cells known as enterocytes. And because the more surface area we have, the better your absorption of nutrients will be, we find that those enterocytes, which are just simple columnar epithelial cells, are covered with microvilli. So we're going to study the microvilli, also known as the brush border, a little bit later, but it is important to recognize here the different layers of the small intestine. Now, in addition to these enterocytes, you're going to see goblet cells secreting mucin. Now, this mucin protein interacts with water and it lubricates the mucosa to allow the food to pass through a lot more quickly. Now, at this point in time, the food is no longer called a bolus. As that bolus mixes with the contents of the stomach and becomes more watery, it's renamed chyme, C-H-Y-M-E. And so that chyme itself is far more liquidy, so you don't need quite the amount of mucose lubrication that you did in the esophagus and the pharynx, but nonetheless, it is there. In addition to all these resources associated with digestion, you're going to find that you have lacteals, which are specialized lymphatic capillaries for lipid absorption. And you can actually see these right here, these little green vessels. And they're called lacteals because as the lipids are absorbed from the lumen into the lacteal, the fluid looks a creamy, milky color, like lactating milk. And so that's the idea there. It's lacteals. Peyer's patches, which you should be familiar with from lab. Those Peyer patches would be found in the ileum, and they would be located here within the mucosa. So what's known here is the mucosal associated lymphatic tissue. That would be where you'd find a Peyer's patch in there to help with immunity. Within the duodenum, you would find duodenal glands in the submucosa. And those duodenal glands, again, only present in the duodenum. I'll remind you is right over here. And those duodenal glands secrete a bicarbonate ion-rich mucus, and that's going to help to buffer some of that very acidic chyme coming from the stomach into the duodenum. So again, this is very similar to what you saw coming here from the mucosal cells of the stomach to protect its own mucosa. And so right here again, you're looking at the small intestine, you're looking at this section of the duodenum, and this is where we begin to tie in accessory organs. So you'll see here the liver and the gallbladder, as well as the pancreas. Now notice, of course, that each of these is going to be able to deposit secretions into the small intestine through what is known right over here through the hepatopancreatic ampulla and sphincters. The ampulla is the opening. It's embedded in what's known as a major duodenal papilla, or a raised region, and it's going to be surrounded by a sphincter. And so that sphincter can constrict and close off the opening, or dilate and relax and open it back up. So we're going to talk for a minute about the liver first. So you've studied liver anatomy quite a bit. It does have the 4 lobes with the falciform ligament separating the right from the left lobes, and the round ligament here as your remnant of the umbilical vein. The gallbladder tends to be situated right here on the inferior side of the liver, and so of course its initial ducts are going to originate right here at the base of the liver. You can see that right there. There's the gallbladder with the cystic duct right there merging with the common hepatic ducts from the liver. So all that happens right at the very inferior side of the liver. Now if you focus in on a histology study or a slide of the liver itself, what you find is it looks very organized. It's going to be divided microscopically into tiny lobules, and each lobule is recognizable by a hole in the very center, and quite frequently a large hole here on the outskirts between lobules that neighbor one another. And all of these structures are really significant in terms of the function of the liver itself. So just to show you right here, each liver lobule is made up of numerous types of cells. The most predominant are hepatocytes. Now, hepatocytes are very important for our discussion in digestion because they are the ones that produce bile. Almost 1,000 milliliters, right? 1 liter of bile every day. But in addition to that, they can actually process blood coming from the hepatic portal system, storing glucose as glycogen, synthesizing proteins, converting toxic things like ammonia into urea, and then of course synthesizing lipids and lipoproteins like HDL, VLDL, and actually using that to help transport excess glucose, amino acids, fatty acids. And so those hepatocytes, again, if you take a look, are the most predominant cell. It's all those cuboid-shaped cells that look brown in color. Those are hepatocytes, making up the majority of what's in each liver lobule. You also have Kupffer cells, and Kupffer cells are stationary macrophages, that's all these tiny purple cells here, that help to eliminate debris or potentially infectious pathogens. Now, when you take a look at the structure of the lobule, you tend to see on the outsides what's known as a portal triad. And the portal triad itself is a collection of 3 vessels. Here you can see a hepatic artery. There we go. Right? You're going to see hepatic portal veins, and you'll see bile ducts. It's important to know the function of each one. And so first off, the hepatic portal vein right there is what's actually relaying blood that was drained from digestive organs through the liver for processing. The hepatic artery, on the other hand, is actually supplying oxygen-rich blood to these cells to keep them functioning. And then finally, you see the bile ducts right here. Right? So of course, when you take a look at the large-scale liver like this, it's important to remember that you have the right and the left hepatic ducts of the liver. Now, each one of those hepatic ducts is actually receiving bile from the combination of many, many bile ducts just like this. So as those hepatocytes are making bile, right, they're sending it through tiny little tubes known as bile canaliculi, which then cluster here to form larger bile ducts. And the bile ducts then transport bile all the way out through the hepatic ducts, so on and so forth. And so a big thing to remember here is even though the liver and the gallbladder are both associated with bile, it's only the liver that is making bile. Right? And so make sure you're familiar with the function of all those vessels within the portal triad. Now the last one to see here is the central vein. Right? There's the central vein. So of course we mentioned your hepatic portal vein receiving blood from digestive organs and filtering it past the hepatocytes. Well, it does that right there by sending blood through what are known as liver sinusoids. And what you can see is that blood is going to run past macrophages, right, those little Kupffer cells, and hepatocytes, being processed, primed, and cleaned before it all dumps into the central vein. And so the central vein there more or less marks blood that has been processed and is ready to return to circulation. And so you see that the central veins here eventually leave, running to hepatic veins and then back to the inferior vena cava. And so please make sure that you know the functions of all these vessels here, including the bile canaliculi, and how they're related to the function of the liver. Right? Now, the gallbladder itself is going to take that bile that comes from the liver, and it's going to do a number of things with it. It'll concentrate the bile by dehydrating it and then store it for as long as it needs to before it's released into the small intestine. The idea here is that, first off, I give you the pathway of bile transport, starting with bile canaliculi, running through bile ducts, all the way down here to your common hepatic duct. Now, this is where the gallbladder pops in to help. So again, we took a look at this image here. Here's that common hepatic duct. And so the liver produces all that bile. It runs down through the common hepatic duct, and it literally backtracks through reverse peristaltic contractions to the cystic duct. And the cystic duct is what holds it, pushes it here into the gallbladder to be stored. Now, chemical signaling, when the time is right, will allow contractions of the gallbladder to release the bile, send it back out the cystic duct, and then down here through your common bile duct, all the way down to the hepatopancreatic sphincter, which, if that sphincter is relaxed and opened, the bile can now run right into the duodenum itself. So the bile itself tends to look kind of yellow because of bilirubin that is present. And of course, jaundice can occur whenever you have a blocked bile duct that then allows this bilirubin to transmit into white or light tissues of the body, including fat tissue. There is going to be a lot of bile salt here in the bile itself, and that's important. But anytime you see salt, that has the ability to crystallize, and that's why gallstones can start to form here in the gallbladder. And this is looks like a painful situation here. In situations like that, sometimes a cholecystectomy is used, and that is the term for gallbladder removal. Now, the other accessory organ that we look at here, and again, we've seen the liver, the gallbladder, is the pancreas. The pancreas is going to function as an endocrine and an exocrine gland, and we're focusing here on the exocrine functions. We'll study pancreatic juice, which would be produced here by the pancreas itself, and then transported down the pancreatic duct, right, all the way here to the hepatopancreatic sphincter, and then also released into the duodenum. So keep in mind, the pancreas is producing secretions into the duodenum at the same time that bile is being sent to the duodenum. The idea here is that once again, that sphincter, that hepatopancreatic sphincter, must be relaxed for the pancreatic juice to enter the duodenum. And we'll study those functions more in 25B. You finish out the alimentary canal with the large intestine and the rectum. Much like the small intestine, you'd still see simple columnar epithelium forming the mucosa, but you don't need as much surface area for absorption here. And so there's no villi, no microvilli. You still do have deep intestinal crypts with a lot of goblet cells, and you'll see this look just like this until you reach the rectum and the anus. And at that point in time, you start to see stratified squamous epithelium once again because fecal material tend to be more dehydrated and abrasive to the mucosa in those regions. The muscularis region here is pretty unique in the large intestine, and I wanted to point that out to you. So, of course, you have the longitudinal and the circular layers of smooth muscle, and the circular layer of smooth muscle tends to tighten at more regions, or at certain regions more than others right here. And what it does is it forms the look of pouches. Those are known as haustra, right there. Likewise, that longitudinal muscle is actually relegated to a single strip that runs here across the length of the intestine itself, and that is called the tinea coli. Right? And so you kind of see a difference there in the development of the muscularis. The rectum itself is an erect or a straight tubule right there that allows for defecation reflexes to occur and is guarded by the external and the internal anal sphincters, which we'll take a look at a little bit more in the second PowerPoint. So that gives you an idea of all the alimentary canal organs and the anatomy of each. Now, all along the way, you might have noticed that there's certain immune functions of all these organs. And so I've gone ahead here and just made a separate slide that summarizes each one of these. We've studied the function of just about all of these different types of structures, only here we've given them some names and more specific locations. So I will ask you to be familiar with the immune functions of the digestive organs as listed here. Right? So that's it for chapter 25A. You'll start on chapter 25B and make sure you're prepared with both of these PowerPoints for our lecture on Thursday.